we talk about, whenever we talk about any GPCR signaling, we mostly have seen, the secondary messenger that is very commonly being used in the trafficking or transducing the signal is cyclic AMP, right? You guys remember we have seen several examples. In case of rhodopsin or phototransduction pathway, of course, we saw cyclic GMP was the molecule that was being governed or controlled. Here in other trafficking, other GPCR pathways, for example, we talked about epinephrine, how it can go on a different cell types, activate the receptor and have some different effect, right? You guys must be remembering about liver cells, muscle cells, what does it do, or in heart cells, what does it do? That was the early classes we discussed. So in most of these cases, it goes and activates, or the secondary messenger is cyclic AMP. Cyclic AMP is made up from what? ATP. So the enzyme which makes cyclic AMP is adenylyl cyclase, right? So for example, when let's say when we have low blood glucose or low blood sugar, and what happens is your alpha cells in your pancreatic islets, they will release glucagon. You guys know about alpha, beta, gamma cells, what do they do? Everyone? Good. Beta cells secrete insulin, alpha cells secrete glucagon, very good, and delta? Very good. So it depends. So whenever you have more glucose, of course insulin is there, so that you can utilize the glucose. And in case of sudden danger, the fright and fight hormone is your epinephrine, right? So adrenal gland will immediately release epinephrine. So in both these cases, both glucagon and epinephrine, they will go and signal liver or muscle cells to depolymerize glycogen. What is glycogen? Yeah, when you have excess glucose, it is polymerized and it is formed glycogen, right? Most of you must have heard this process, no? It's very commonly which happens. What they do that they will depolymerize glycogen, because you need glucose, that's why you want to break it down. That's your kind of storage source. Now in liver, what happens is glucagon, epinephrine, they bind to different GPCRs, and both receptors will activate the same G alpha subunit simultaneously. In your liver cells, when you activate glucagon and epinephrine, both of them they will go and bind to different receptors, but they activate the same G alpha S subunit, which activates essentially the adenylyl cyclase. This adenylyl cyclase will induce the cyclic AMP, and then cyclic AMP will go and give some metabolic response, whatever is expected out of that. So this is one example where two different receptors being activated by different ligands, but going and binding or activating same G alpha S. G alpha S is stimulate. So the G alpha S is mostly going to do something stimulation to that. However, in adipose tissue, you can see here you have this receptor which can be activated by your epinephrine, glucagon or ACTH. What is ACTH? Very good. So these guys can go and activate your receptor here. This receptor will go and associate with G alpha S. When it associates with G alpha S, it is going to go and activate adenylyl cyclase. Whenever your adenylyl cyclase is activated, there will be more cyclic AMP generated. This more cyclic AMP will do some intermediate activation. The end point is that the breakdown of triglycerides to fatty acids and glucose. That's the lipolysis will happen. At the same time, in same adipose tissue, you have PGE, which is prostaglandin receptors, or adenosine. These guys are inhibitory. What they do? They go and associate with the G alpha I coupled or G alpha I G proteins, which will go and inhibit adenylyl cyclase. So you can see that in one place two different hormones, two different receptors doing the same job of activating same G alpha S. So there is same response. In this case, there is maybe you can take it as a homeostatic balance, right? One pathway is activating something, another pathway is trying to suppress it, so that you maintain the balance of lipolysis. Is it clear? You guys are following? This could be a very common mechanism in most of the cell types. You might have seen feedback loop or something, but this could be another way how you can govern the signaling output, where you have a receptor. I'll give another example towards the end of it. You have a receptor which will activate cyclic AMP, so that it will induce the lipolysis. At the same time, you have another receptor which is gonna inhibit adenylyl cyclase, lower down cyclic AMP, and maybe inhibit the lipolysis. So these are the different examples what happens. So now you guys must have gotten an idea that we are talking about cyclic AMP and adenylyl cyclases, right? So adenylyl cyclase is the enzyme which is gonna convert what to what? It will make cyclic AMP. So this was the science paper published in 1999 and 1997. You see that science paper, the crystal structure of catalytic domains of adenylyl cyclase in complex with G alpha S, and this is when they have used the non-hydrolyzable form of GTP. You guys remember we did talk about this. So this is when the application of this non-hydrolyzer form will come, where you render that G alpha S in constitutively active mode, because you want it to go and constantly associate with the effector, that is your adenylyl cyclase, and then they try to get the structure of that. So it's very interesting. If you look at the structure of this adenylyl cyclase, again it's a multi-pass transmembrane protein. You see this has these two different modules. You have M1. It is not like your GPCR. In case of your GPCRs, your N terminus is always extracellular, C terminus is in intracellular, right? But these guys are slightly different. What they are doing here, your N terminal is also intracellular. It is not exposed outside because it's not a receptor. There are other reasons why it could be. Then you have M1, where you have this transmembrane regions are inserted inside the membrane and they are associated with it. The C1 is the C terminal, so it has two C terminals they have designated C1, and the other part of this is M2, which is similar to M1, and it also has the C2. So phospholin is a molecule, is a chemical that goes and activates adenylyl cyclase. So whenever you want to increase cyclic AMP levels in the cells, you add phospholin to that. So phospholin goes and binds to this C1 and C2 domains, and it activates the adenylyl cyclase. Whereas it has ATP binding site, then there are magnesium ions which are very important to stabilize the ATP binding. So that's how the structure of this adenylyl cyclase which we know today. So this guy, this person, the John et al., they were the first people in 1997, they got the structure of this. Now when you look at this structure, what do you guys think? Would it have been easier for these people to crystallize this protein? No, Zoya? What do you think? What would they have done then?
Since it is a membrane protein, first of all, therefore crystallization of such proteins are difficult. So I think they have used some other technique like cryo.
No, 1997 there was no cryo, no? Cryo came now. If let's say we have to do this, what do we think? What can be done? Aman? I understand Zoya's point is valid. It's a membrane protein. Maybe it's a multi-pass membrane protein, right? It's very this structure we know today. I mean, I'm just reminding, it's 1997, ages ago. And then they followed it up 1999, there was another Nature paper. So that's the beauty of it, no? You dissolve something and then it's Nature or Science. It is not easy to get anyways. So what do you think? What one can do if you, let's say Teja, you are go back 1997, you might not have been born maybe. No? Good. Okay, so we cannot ask. Let's imagine I have to go back to 1997 and do this experiment. What can we do? They must have tried all permutation combinations. You guys remember we talked about Brian Kobalka, and then he was trying to say how did they crystallize beta 2 adrenergic receptor with G alpha S, and then they really understand what are the structural changes which are happening over there, right? So now the question is what one could do. Saheli, do you have any ideas? Yes. The nanobodies or something like that. Then that was one of the ways. Okay. Do you have any idea, Aniket? Okay. So you remember these were those conventional crystallography days, right? So what these guys do, the advantage what they had was if you take only this M1 or this half fraction, it is soluble. If you take the other half fraction, it is soluble. So what these guys do, they purified these two fractions and then they put them together along with phospholene and your ATP. Then in vitro, the whole adenylyl cyclase they were able to reconstitute like that, which is functional one. They must, of course, they must have tried a lot of other methods, but this is what worked for them. Did you guys, did you, huh?
This is the correct structure that what they wanted to see.
Huh? No, see, most of the time, even most of these, even if you take G protein coupled receptors, you can still express them in bacteria. And then you stabilize them by using some detergents and all of that. So that people do very commonly. That they do commonly. So maybe to get it really functional in terms of making adenyl cyclic AMP, maybe you put them on some membrane discs and all. So that they did not explain how did they do it, but what I read is that you have two fragments, you put them together in a tube, provide phospholene, and then so the idea is even if you have this membrane associated amino acid fragments or amino acid regions, still they were soluble proteins. So when you express a protein in bacterial cell, right, you take any protein, you take the sequence, clone it, put it in bacteria, induce it, and then you purify, right? That's how you express it. It comes in a soluble fraction, meaning it's not getting precipitated or it's not going into inclusion bodies and all that. So I think they got lucky where they were able to get it in soluble fraction individually, not a full length protein, but then you purify both fragments and put them together. It's kind of, you know, they are forming a homodimer kind of thing, but they are not. They express the recombinant protein they made. Which one? Phospholene. You do that. That's what you do it. That is doable. They did it. So that's kind of reconstitution of the whole complex they did. So phospholone is, it's not endogenous, it's a chemical compound. So you add it and it goes and binds there and somehow, and it is like, it amplifies the activity in many fold actually, that people use very commonly in experiments.
But did they follow up this with studies on membrane environment?
Oh, that I don't recollect. I think, that time, if I recollect something, they just resolve the structure of it. They did not do many more than that.
Any of these membrane proteins which you are trying to produce by recombinant technology will be expressed in the cytoplasm and then
No, you will struggle. You will find so many proteins, you will not be able to express them. They will go to pellet. I mean, so there are different systems you can express them, right? I mean, this is for everybody. If you guys remember, we were talking about affinity chromatography, you tag a protein and all that stuff, right? If you have a G protein coupled receptor, for that matter, if you have any membrane protein, the one of the major caveat or drawback these membrane proteins come with, that they will have hydrophobic, significant amount of hydrophobic amino acids, right? So what point he is trying to make it, there is always a possibility that they will aggregate together, right? So so as I said, we have multiple expression systems. Whatever these kind of stuff you want to do, if you want to get the structure of a protein, you need pure, B, you need more amount, so that you can actually try to crystallize or visualize and all that stuff, right? So you need quality as well as quantity over there. How can we make more and more protein? There are multiple systems which people use to express them. You can use bacteria, which is our E. coli. You can use insect cells, which are SF9 and other insect cells. The advantage of insect cells is that the proteins will have all the post-translational modifications as well. The disadvantage of using bacteria is that proteins will not have any. Gaurav, if you take a protein and try to put it in bacteria and make a recombinant protein, what would be the disadvantage of that? They do not have post-translational modifications, right? In fact, when you do, this is good if you have A protein, B protein, and if you want to show that this A, B protein binds to each other, you express it in bacteria. But the moment these A, B proteins need additional factor for interaction, that you cannot do with bacterial proteins, express proteins. The moment you need a post-translational modification, we cannot do it. So that is the limitation of that system. So people have used SF9, insect cells. In insect cells, you can get post-translational modifications. Now people are moving towards mammalian cell cultures. HEK293T cells, you must have heard, no? They are called as protein factories because they can have extra chromosomal or episomal, because they are transformed with sp40 large T antigen. There is episomal replication of the plasmid can happen and you can get ton of protein. So now people are employing those methods. So those days, I don't think these methods were evolved. So they might have followed a conventional method. Now to purify these membrane proteins and all, there are nanodiscs and whatnot. So many things have come. But I think these one, so you can imagine why Brian Kobolka got, for resolving a structure of beta-2 adrenergic receptor in active state. That's the major work what he did, and he got a Nobel for that, right? Because nobody ever did that. So those were those days when it was not possible. Today, there is a person called Ashish Manglik. He was a postdoc with Brian Kobolka, who published a cell paper. Eventually, he moved to UCSF, and then he resolved the structure of some of the G protein coupled receptors, and he is using those nanodiscs and whatnot. So the times are evolving. So with every system, you will be able to make the different, better version of what you see inside the cell. So you can reconstitute that. There is a complex called, have you guys ever heard about MCM helicase complex? Mini chromosome maintenance complex? No, Zoya? Have you ever heard about it? Yes? Pratibha, have you ever heard about MCM complex? Do you guys know how the origin firing happens? How the DNA replication starts, right? So you have, there is some origin recognition complex, right? That's, and DNA replication happens both the sides. So this MCM complex is same like this, but it has 6 proteins, right? And they are sitting, the 6 protein ka as a ring hoga, they are sitting right like this, and the firing will happen and they will start moving like this. Though are helicases, they have ATPase activity. They are the ones who will unwind the DNA. So they will start moving both the side, right? People have reconstituted the whole assembly in a tube by purifying each protein. If you can do that, this is not, it's not impossible. So the point is, we do not understand the power of a system unless we don't try it ourselves. So they got lucky where they got 2 fragments, put them together in a tube, put phospholin and this ATP and all those things, and they were able to reconstitute the structure. I don't reconnect, but they must have definitely done the functional assays where you can measure the cyclic AMP production when you have this assembly, or you do not, when you remove the ATP, whether it happens or no, and all those checks they must have done. To answer your question, they must have checked whether this assembly is definitely making cyclic AMP or no. So if, yeah.
Still, if you have any membrane protein which you are producing by recombinant DNA, you change the vector also, like make it insect or whatever, then everything will be trafficked ultimately into its membrane, and then it has to be extracted.
Yes, you can solubilize them.
Solubilize
Yes.
detergents and extract
Yes.
it from the membrane. So it is not right that we are taking it from the solution.
No, I'm not saying solution. What I'm trying to say that soluble fraction, meaning when, so maybe I was not clear. So what I'm trying to say when I say soluble fraction, meaning it's not the cytoplasm. What I mean by soluble fraction is that when you take, let's say you take bacterial cells, you lyse the cells in certain buffer, right? The buffer composition will have all the required stuff which will make the protein soluble, that will help to extract it in from the lipids or whatever it, that's how it will shield it. So that's what I mean, and then you get it in soluble fraction. Soluble fraction and pellet fraction, I was talking in terms of, you take the bacterial cells, Vaishnav, you lyse them, okay, you put them on centrifuge tube, you spin them at very high speed, then there will be something which will settle down, something will be in the buffer. That whatever something is the buffer, that's the soluble fraction I mean. And whatever is settled down, that's your pellet fraction. So what I was trying to say, not the cytoplasm, the soluble fraction will have everything extracted in it, and then you get it. Is it clear? So you guys will do these experiments very soon. I don't know, some of you must have started doing, but you will do it. If you start looking at it little bit more carefully, you can see this is your M1, this is M2, and now they are showing this G alpha S. This is the paper which I got this structure from. You have this G alpha S. You guys remember we talked about G proteins and how do they get activated upon binding to GTP versus GDP very early. There is a switch region, switch to region, there is a magnesium ion, and then these 2 molecules move like this to accommodate the magnesium. You don't remember? I think you must remember the figure, no? You have switch 1, switch 2 regions, there are magnesium ions, then you have GTP in the pocket, you stabilize that phosphate by bringing these 2 amino acids together. The moment GTP hydrolyzes, it gets into open structure. Right? I have sent the slides. If you don't find, just go back and check it. So the point is, in this case, again they are trying to show the importance of this switch 2 region in the association with adenylyl cyclase, because your G protein will associate with adenylyl cyclase Tanvi only and only when it is GTP bound, right? Which means that, Shivam, are you following what I am saying? Does it sound like Hebrew? Are you guys lost in between, or you are still with me? So the point is, you guys remember the basic of GPCR signaling, what is happening? You have GPCR, shine, then what happens? Ligand comes and binds to it, then? How? Right, then what happens? There is a conformational change. Conformational change you should not forget, right, Aniket? It's very important. Then it separates, right? Good. Then it goes and binds to the effector. In this case, it's adenylyl cyclase. In your phototransduction, what was it? PDE, the gamma subunit of that, right? So in this case, it's so you guys definitely remember that G alpha S or G alpha subunit will go and bind to the effector protein, right, Rishika? Only and only when it is GTP bound. So that's when the importance of your switch region is coming, because when it is GTP bound, the switch region has certain conformation. When it is GDP bound, it moves away. That's why the first classes were also important. So here they have tried to do these simulations. I think I don't remember whether it's the correct paper, but there must be definitely these structures are also should have been solved. So they have showing that you have phospholene, which is associated with your C1 and C2, which are the two these domains over here. Then you have switch to region of your G alpha S, and that's how they associate. So G alpha S associates with adenylyl cyclase, and then essentially it will lead to its activation. Whenever it gets activated, it will make more and more cyclic AMP. Is it clear? Now, we all of us we know we have a GPCR, you have a ligand. Let's say epinephrine goes and binds to the GPCR, activates it. When it activates, now we learn that it is going and activating the adenylyl cyclase. Once it activates adenylyl cyclase, it is making cyclic AMP. What does this cyclic AMP do? The whole purpose of we studying all this mechanism, you remember in phototransduction pathway what happened, Aman? The ultimate thing what happened is the ion gated channels were closed, right? That's what happens. In between there are 100 steps, no, like वो activate G alpha T activation, then PD gamma subunit को sequester करो, right? Then alpha beta goes, then it converts cyclic GMP to GMP, lowers the concentration, gate closing happens. In this case, you have a GPCR. We give an example of epinephrine, right, Muskan? So you have epinephrine which is present, then your ligand comes. Sorry, epinephrine is a ligand. Epinephrine goes, binds to the GPCR, activates cyclic AMP, right? Now what will it do? What that cyclic AMP does? You guys remember we talked about this particular thing? Go back. Here. In liver cells, you have epinephrine which goes and binds to the GPCR. Then GPCR active, not this, sorry. Here, you take this adipose tissue example. You have epinephrine goes and binds to the GPCR, activates adenylyl cyclase, makes more cyclic AMP. What is happening? It leads to the breakdown of lipids. So that's the activity is happening. No, that's the signal come from outside. Hey, this is the time you should induce lipogenesis. It got transferred. So cyclic AMP informed the intermediate. Now what is that intermediate that is gonna do this job? What do you think? What do we know? pKa, protein kinase A. You guys know about this pKa? Everyone knows? Then I need not to tell. So protein kinase A is also called as cyclic AMP dependent protein kinase. Of course, pKa has two subunits, regulatory subunit and catalytic subunit. There are two catalytic, two regulatory. So essentially when you have pKa which is inactive, these catalytic subunits somehow inhibit the regulatory subunits. Regulatory subunits inhibit the catalytic subunits. Whenever you have cyclic AMP in the system, cyclic AMP goes, binds to regulatory subunits. The regulatory subunits will dissociate from the catalytic subunit. Catalytic subunit is free to go and do whatever it wants in the cells. So the mechanism of this activation is very interesting. Now everybody can see this? Good. So if you look at this, so this is where your regulatory subunit is. This is the dimerization domain where two regulatory subunits are dimerizing now, and these regulatory subunit have these cyclic AMP binding regions CNBA, CNBB. This is where you can see that there is this tiny red color molecule which is sitting inside, that is your cyclic AMP, right? These are the cyclic AMP binding domains. The most interesting mechanism how this regulatory subunit inhibits catalytic subunit is that these regulatory subunits, they have a pseudo substrate domain here that binds to the catalytic subunit. So, the pseudo substrate domain will go and bind to the catalytic subunit in a way that catalytic subunit cannot phosphorylate anything. That is how it keeps it in inhibitory state. Okay, Gaurav? Sorry? Yeah. So, your regulatory subunit have different domains in it. First, it has dimerization domain, so that it can dimerize with other regulatory subunit. It has cyclic AMP binding domains where cyclic AMP goes and binds, but then it has a motif, a domain, a motif, let us say motif, that is called as pseudo substrate domain that binds to catalytic subunit. Pseudo substrate domain. Pseudo substrate domain meaning it is fooling the catalytic subunit that this is the substrate. Not yet clear? You understand it? Pseudo substrate meaning it's not a true substrate, right Shivam? It's not a true substrate, but it associates with your catalytic subunit and kind of telling catalytic it is kind of mimicking the substrate, so that catalytic subunit will not be active, it will not do anything productive. That's what it is doing. So, the moment your cyclic AMP goes and binds into these cyclic AMP binding regions, now you can see that, so you can see the conformational changes. You have cyclic AMP binding. So, here when you have inactive inhibitory conformation which is shown in this green color, whereas there are these amino acid residues which it is engaged with. The moment you add cyclic AMP, there is a conformational change which happens in this region. That is how this will remove the inhibition from the pseudo substrate domain, so that your catalytic subunit can dissociate and go and bind to its actual substrate. Is it clear? So, in general, this could be mechanism to any proteins. You guys can understand, right? You have a protein which is in close conformation. You can say that this is close conformation which is kept, so that, and okay. So, when you say that a protein is kept in a close conformation versus an open conformation, proteins are made up of amino acids, right? So, the way the folding which happened, the way these amino acid residues and their R chains are oriented, you guys are following everybody what I said, right? You guys know, can you tell me 5 amino acids? Anyone? Then we tell 2 amino acid names. Which one you like? Arginine and lysine. Okay, positive. She is very positive person, so she like positively charged. Anybody else? Disha? Lysine, glycine, proline, valine, alanine, tyrosine, phenylalanine, tryptophan. I like tryptophan actually. It has very nice ring, no? So, gluten. So, there are so many of them. Now, you imagine you take this tryptophan. It has a ring. This ring, when you say that this protein is in inactive state, the orientation of this ring could be different. The moment you, let us say, add ATP over there, whether this is going to make some connection, some bonds with this, this ring can move like this, which will lead to some conformation change, then it could say that it is in open conformation. Same thing is happening over here. The moment you add your cyclic AMP, these amino acids are trying to make connection with it. So, there is a change in the conformation. That is what it is depicted over here. Actually, this picture is not extremely very clear to me, but these are the 2 amino acids, arginine 209 and glutamine 200. These are the 2 which are engaged with the associating with the cyclic AMP, which is causing the conformation change, and that's how the activation of your pKa is taking place. So you guys should remember three things. pKa, protein kinase A, has two subunits, two regulatory subunits, two catalytic subunits. Now what you are trying to understand, how pKa is activated by cyclic AMP, right Saheli? So cyclic AMP goes and binds to what? Regulatory subunit. So how your regulatory subunit is inhibiting catalytic subunit? Pseudo substrate domain, then that's how it mimics it. The moment you add cyclic AMP, there is a conformational change happening, then they get separated. So your catalytic subunit can do whatever it is supposed to do. Is it clear? Good. Allosteric. I do not know whether you can call it, I mean it has two subunits, regulatory and catalytic subunit. I do not know you can call it self-allosteric as of yet, because I would imagine if you have same protein which has different domains and those domains are changing, then I could say that self-allosteric. Maybe here it is more of some other sub-protein kind of regulating it. You understand? Because this is not the same protein. Catalytic subunit, regulatory subunit, these are different subunits. I don't think that is the same protein. They are multi-protein complex coming together. You see that these are the two subunits, and then, so I don't think that's the same protein. If you have the same protein, then maybe that can happen. Okay, so for example, we will take one example of gluconeolysis, which is opposite to glycogen synthesis, right? Gluconeolysis is nothing but the breakdown of stored glycogen. So whenever you have glycogen, there is this enzyme called glycogen phosphorylase. Now you guys will have to little bit strain yourself, because it's little bit complicated pathway and there are too many molecules. The concept is very simple, but there are too many names. So just shake yourself up little bit and then focus on it. If you still have questions, Rishika? Okay. So you guys know, right, glycolysis, what happens? There are too many intermediates. You have glucose, then glucose 6-phosphate and this and that and all. So every time there is this kinase that comes. So that's how you will have so many names over here now. Brace yourself, Vaishnav. So you have glycogen. This glycogen phosphorylase is an enzyme that will phosphorylate that will convert this glycogen into glucose 1-phosphate. That is something very simple. So you have epinephrine goes and activates the GPCR. GPCR, once you get activated, activates G alpha S. It goes and activates adenylyl cyclase. Adenylyl cyclase activate pKa. pKa will go and enhance the conversion of glycogen to glucose 1-phosphate. So now when you want to convert glycogen to glucose 1-phosphate, or when you want to have this glycogenolysis, that is the breakdown of glycogen, it can be done two ways, right? If you want to, let's say you have molecule X which is there in the cell, you want to break down that molecule. There are two ways to make sure that this molecule can be broken down, or you can induce the lysis or breakdown process of this molecule by two ways: by inhibiting its synthesis and by stimulating its breakdown, right? The same two way, प्रोडक्शन भी कम करो और उसको खत्म भी करो. So your epinephrine pKa does the same thing. It inhibits glycogen synthesis and it stimulates glycogen degradation. Now this is the complicated pathway that I was telling you. Let's focus on the upper part of it. So pKa, which is active, what does it mean when we say pKa is active? So there was cyclic AMP went bind to the regulatory subunit में conformation change happen, it got separated, your catalytic subunit is free. This is your active subunit. As we said, it is going two things. First, it is stimulating glycogen breakdown. So what it does, it goes and activates. So there is a kinase which is called GPK. I forgot the name of this kinase. The side में लिखा, sorry, glycogen phosphorylase kinase. It's difficult to remember the names. So glycogen phosphorylase kinase is over here. So you guys, we talked about it, right? Phosphorylation can be activatory or inhibitory. In this case, it is activatory. So what it is doing? You have inactive glycogen phosphorylase kinase. Thus, PKA is a kinase. Remember that, right? So the kinase will go and phosphorylate a substrate. What is the substrate of PKA now here? GPK. This GPK will be phosphorylated by PKA. That is not the direct substrate which is doing anything to glycogen. So this GPK which is activated will go and phosphorylate glycogen phosphorylase and activate it. Now this GP, which is your glycogen phosphorylase, will go and convert glycogen to glycogen 1-phosphate. That is one arm of it, where it is doing indirectly, not directly to glycogen phosphorylase. The other arm is that you inhibit glycogen synthesis, where you have active glycogen synthase. So you inactivate that. So essentially you take glycogen synthase, phosphorylate glycogen synthase. Whenever it is phosphorylated, it will get inactivated. It's very simple mechanism. So you guys can imagine actually that you have same pKa molecule, but how many substrates does it have in the cells? Right? It's already in front of you, there are 2, 3. So you can imagine it will have, I don't know, 200, 300 different substrates it might have. So that's how the signal amplification and transduction can happen. Of course, it's not random, right Teja? It has to have a biological context to it. It's randomly, ऐसा ही तुम्हारा पीके चलो अभी मन हो गया तो अभी एक्टिवेट हो गया, अभी हो गया, ऐसा नहीं होता. There is a biological context to it. What is the biological context here? Epinephrine going and activating glycogenolysis. Your glycogen stored जो है उसको break down करना है. You need more glucose. You are low on energy. That is the context. Yes. You want me to repeat the questions? Her question essentially is that you have whatever XYZ ligand is coming and binding to any receptor GPCR, right? Let's imagine you have one GPCR, you have one ligand coming and binding to it, or you might have so many GPCRs coming and binding different, but everything ultimately going and binding down to adenylyl cyclase. This one adenylyl cyclase is going and activating one PKA, protein kinase A. So how does this protein kinase A know that I have to do different function when a different stimulation is coming? Is it the question? Good. I understood the question. Yes.
Even PKA, if when coming to downstream signal, if it is activating, so the phosphorylation does not actually only lead to its activation. So in the other case, phosphorylation of a protein leads to its inactivation. So that is how it can dynamically activate one protein and inactivate.
No, it can do. That is not the question. What she is asking is, how, let's say this is happening in adipose tissue. Let's say you have liver cells, you have adipose tissue. Let's say you have pancreatic cells. Then you have, pre-adipocytes. There are different examples, right? So I am partly answering her question as well. So every system, every cell will have certain set of G alpha S molecules, certain GPCRs. The proteins are same over there, right? So ultimately your peak. So I'll give one example. You have this liver cell, for example. or you have your adipose. Adipose tissue which is already mature adipose tissue. And if you have an example of during the differentiation of adipocytes, in all these cases your G alpha S is coming, that is true. So, you remember we threw some numbers, no, there are 20 different G alpha S which are present. Now, imagine from the brain to any other tissue, there are so many different cells, so many different tissues, there are different G alpha S's. In case of adenylyl cyclases, however, there are different types of adenylyl cyclases. Adenylyl cyclase 3, adenylyl cyclase 5, adenylyl cyclase 6 and several other. So, ADCY3 or adenylyl cyclase 3 you can find only in the neuronal cells. Adenylyl cyclase 6 you might find in pancreatic beta cells or adipocyte cells. Still there is a limited number, that is fine. So, even if they are more than one different adenylyl cyclase, they express tissue specific expression is there. Okay, some of them they will express only and only in brain or neuronal cells. Some of them they will express only in the hepatic cells. But again there might be overlap between their expression. That is one thing. Second thing is, even if you have, let's say even your adipose tissue, you mostly work with your lipid metabolism, right? Whether you break down the lipids or you synthesize the lipids. Like for example, what we saw in lipolysis. Or if you have in liver, let's say you are working with your glucose metabolism. Here mostly you want more glucose to be released in the system, or you want to inhibit the glucose. You have more glucose, so you make glycogen. And in case of differentiation of adipocytes, when you have pre-adipocytes, now it's a completely different phenomena where you have pre-adipocytes from multipotent stem cells. Pre-adipocytes will come from pre-adipocytes, they will mature into adipocytes. That this whole process is happening. This also is dependent on GPCR signaling. So, every time you have, there is a different cue which is coming, a different receptor which is coming. So, the same different receptors will get activated with different molecules or different ligands, right? Let's say you have 1, 2, 3, 4, 5 different receptors. So, these set of cells will express very different set of GPCRs than your liver cells, right? Liver cells will have different set of GPCRs than your adipose tissues. If you take your neurons, those will have a different set of GPCRs than your secondary metabolic tissues. So, first of all, there is a differentiation of what kind of GPCRs are expressed in different tissue systems. That is dependent on the requirement of that tissue, right? If your brain has more perception related or hypothalamic neurons, they are the primary site of body weight homeostasis. They have a very different set of GPCRs which are gonna regulate the functions than what is present in your adipose tissue. Okay, I'll give one example. Free fatty acid receptor 4, FFAR4. This FFAR4 it will find it in adipocytes only. It will never express in your brain neuronal cells. However, melanocortin receptor 4 is expressing in your brain cells, not in adipocytes. That is first level of differentiation happening. But again, they will go and activate the same G alpha S. But in this case, even though if you have same G alpha S, the biological context is completely different. The requirements are different. So, there is a subset of proteins which are substrates of this pKa. Everything is boiling down to pKa, right? But pKa will have a very different substrate in your maturing adipocytes, whereas it will have very different rate of substrate in your liver cells. So, even though, so that's how the systems are tuned in a way that whatever, that's the first, if you recollect some of the first discussion we had, same GPCR, different cell types can have same defect, same effect or different effect. You remember we saw two different examples, right? Epinephrine can go and activate, we saw that acetylcholine will have a different effect on your heart cells. It will slow down the heart rate when you release acetylcholine, but if you go and put it in a smooth muscle cell, it will lead to the contraction of muscle cells. You remember? So same neurotransmitter, same receptor, it has a different effect. That's when you have the tissue specificity. That's how it is designed. So even if you activate the same receptor, it is gonna go to the same adenyl cyclase. In case of neurons, this at the end of the day you will make cyclic AMP. Cyclic AMP will go and activate pKa, because pKa is same everywhere, but that pKa has a different substrate in a different cell type. That's what it designs. That's how the so if you, I do not know if you take a pKa from liver and put them in adipose tissue, whether it will do the same phosphorylation of other molecules or not. We don't know. So the tissue specificity comes over there. Another aspect is this pKa, what we talk about it, right? It's the same kinase, but we never talk about what are the accessory proteins which might be important, which will function along with pKa to drive that function. Maybe in adipose tissue it will have some specific factor which are specific in adipose tissue, which will activate, which will enhance the activity of pKa, or they will act as a adapter proteins to bring the substrate near pKa. There are too many permutation combinations which will design. At the end of the day, I think it based on your tissue specificity what it goes. Right. So see again, this is its distinction is there, right? When you talk about cell proliferation, cell survival, or cell growth, there is a different set of receptors which will function over there. It's not necessary you have GPCRs only doing that function. You have receptor tyrosine kinases, you have growth factor receptors. That's how the division has happened. So the some set of receptors will do only that job. If you take you take PI3-AKT pathway, you take mTOR pathway, you take PDGF, growth factor receptors, receptor tyrosine kinases. They will do mostly functions related to cell survival, cell proliferation, cell growth. This is what it is doing. You will find few examples where a GPCR is going and activating mTOR to do protein synthesis. But you will always find that PI3-AKT will go and activate mTOR pathway to make more protein, because that is their job. So even if the same adipose tissue has doing different functions, the different receptors function in different steps. Is it clear? Yes. So that's where the substrate specific, because they have different substrates. No, substrate means what comes downstream to the kinase. For example, you have pKa here, right? This pKa, this everything is happening in your liver, right? That's what liver only, no? Anusha, what is that? Yeah, I think it is somewhere it is happening. I'll figure that out. But imagine in this case what is happening. You have pKa. What is the direct substrate of pKa over here? Is your GPK, that is glycogen phosphorylase kinase, or GS, what is glycogen synthase. So if this pKa has these substrates in your liver, it is not necessary that pKa will have same substrate when you take it and put it in your neuronal cells, or neurons, or some other epithelial cells. It's not necessary. Could be. What, let's say for example, you take liver, which is active metabolic tissue, right? You take your pancreas, which are also doing some glucose homeostasis. In both the see again, you have to understand it a bit more physiology. Liver has a very different metabolic function than your pancreas, right? They mostly do secretory functions. But their pKa will have very different substrate that will go and phosphorylate something which is important for secretion of insulin granules. That will be the substrate for that. In this case, it is mostly storage. You want to either break down glucose or store glucose, then you are working at that level, then you have very different substrates. So the set of substrates which are there in different tissues which are downstream to these kinases will determine the final output. Yes, that's exactly what I am trying to tell. So the point is, even if, so what do we think in our cells as of we are talking, we think that only at a time only one pathway is activated? Never, right? Everything is there. Still we have a very good distribution of regulation. No? The whole reason is there is something in between that will determine whatever upstream signal is coming. Even think everything is boiling down to the same molecule, but this same molecule, that's when I said the association with the next protein molecule will determine whether it is supposed to do and not. Even if you take a cell, hypothetically, stimulate with vasopressin, stimulate it with your epinephrine. Both of them, they will not give to the phosphorylation of same protein. In terms of you want to have some activity at the end, right? Vasopressin is doing completely different biological activity than your epinephrine. Which is it clear? Let's understand that. Let's say vasopressin is doing something and epinephrine is doing something else. In both the cases, ultimate end product is very different. So even if pKa is in between, pKa what is going to phosphorylate the next, this pKa might go and associate with completely something different than this pKa molecule. It's not, at a time there will be 1000 pKa molecules which are active and roaming around. But with what molecule it is going to associate to phosphorylate, that's what it decides. That is exactly the mechanism. So the mechanism is, I mean, I do not know in specific context what could be the mechanism. So for example, so let's say you have pKa. So here imagine you have your epinephrine, this is your vasopressin. These both guys went and activated pKa, right? You don't know these pKa getting signal from where. But if you activate epinephrine, it is going and let's say it is going and it's going and doing the expression of gene X, for example, okay? This is the job of your epinephrine. At any given time, your vasopressin is never going to do this, right? Is this clear? Which means that even if this pKa getting the common signal, this protein X, gene X, expression of gene X is dependent on what? The expression of this gene X will be dependent on some transcription factor. This transcription factor will be either in cytoplasm or in the nucleus. Along with this transcription factor, there will be another factor. Let's say this there is a transcription factor S. Sorry. Let's say you have transcription factor TF. Okay. It has to be sequestered in the cytosol, right? Even if you imagine that this transcription factor is the common factor between both of them, vasopressin, epinephrine. When this transcription factor is sequestered in the nucleus by factor Y, that is the only time pKa will phosphorylate this. So that when you are, so that this will be inhibitory phosphorylation, Y and transcription factor dissociate, transcription factor can go and sit on this and drive the expression of X.
So in that case, this factor Y should be elicited by something else.
That factor Y become very specific in context of your epinephrine. So these factors will be there, they will determine. It's never that there will be so many scaffolding proteins, there are so many substrate recognition proteins, there are so many adapter proteins. These are the proteins that do the job. You guys have heard about Wnt signaling? Everybody has heard about Wnt signaling. Is there anybody who doesn't know Wnt signaling? You don't know, we will talk about it, but Wnt signaling is very important during the development, right. So, there is a kinase called GSK3 beta. This, she must be knowing, I don't know how many substrates this kinase have, 400, 500 substrates it will have. But if you activate a Wnt signaling, the only thing it will do, it will, if it is in the context of Wnt signaling, it will go and phosphorylate your beta catenin. That is the only job it will do. In case of Hedgehog signaling, same GSK3 beta will go and phosphorylate, what is that? Gli, which is another transcription factor. But when it is phosphorylating beta catenin, there are 1000 other proteins that are associated with that beta catenin, which should determine that this GSK3 beta is very specific in this context. So, there is a whole layer of another regulators which come, which we are not talking about. And those are the proteins most of the time will have some of the other regulatory role. Otherwise, it will become chaotic, no? Yes.
That key and coming to back go to which substrate and activate.
No, I don't see.
Or are the next level PKA cascades they are different things?
See, the way I understand is, some of these PKA should be kind of a housekeeping kinase which is there. So, the amount of PKA molecules will be always excess than what you need it. So, you have to see, we talked about signal amplification, right. So, that is going to come here also. So, the number of PKA molecules which are being activated will depend on the amount of cyclic AMP which is coming down, right. So, if a job of PKA is to take the signal from top and transfer it to bottom, cell make sure that there is sufficient PKA in the system, otherwise the signal transduction never happens, right. The sensing and maintaining the right amount of protein in the system is very critical, that cell will make sure. Maybe some of these molecules, to answer your question, let us say your E is there, right. This E goes and activates some transcription factor. This transcription factor goes and lead to the expression of so many genes. If at all we want to maintain the right amount of PKA, it is always possible that the same transcription factor will lead to the expression of PKA also. And this, the level at which these transduction pathways work is, which is extremely fast, meaning the cell will make sure always that you will have sufficient PKA molecules there, because this E is going to constitutively activate transcription factor is going to constitutively drive the expression of PKA will be there. Even if you have initially 100 molecules, upon activation you can have 1000, 10,000, 100,000 molecules, because this transcription factor is itself is driving it. Till one of these PKA, one of the other kinase will come and phosphorylate this, so that the Y can go back to the transcription factor and drive it.
Suppose I am PKA, either I got from E or from V. Now I have to go and deliver the signal downstream to the substrate with some more cascades. How could I be sure that I will be taking the right signal to the right substrate?
That will be determined by Yash. Sorry, Jay. So, that is in case of vasopressin, in this case of epinephrine. So, that will depend what is your surrounding. See, most of the time we think that these molecules are talking one on one. It is not always the case in the cell. There is always the extra layer of regulation. So, at the same time your PKA is coming, but it goes and associate with some other molecules. That is how you direct the phosphorylation. Second thing is, even if in the same cell there are this one, the crosstalk will never happen. Another thing, the most interesting part, whatever she was telling, you have two GPCRs which are activated by the same molecule. That, I mean, we will talk about the desensitization. If this G alpha S is constitutively activating another PKA, there is a possibility that this PKA can go and inhibit the GPCR activation itself. That will kind of a feedback loop. So, it is always regulation will be there. So, your pKa is a single molecule, but that pKa, that protein will have very specific substrate, and the substrate specificity could be determined by multiple level. Either you have very specific, either you need an adapter protein, or that the substrate protein might be modified, or the substrate protein might be associated with another protein. So there are too many combinations which will determine that.
You say at that level, the pKa when it gets the signal from E and B, that is same. That is not any different.
So there you cannot distinguish, because as Shritama was telling, even if you have, so that's what I said at the beginning actually here. In liver, glucagon and ephedrine will bind to different GPCRs. Both receptors will activate the same G alpha S protein, right? The same G alpha S will activate the adenylyl cyclase, but here they will induce the same metabolic response. So most of the time what happens is that it again you go back to biological context. See, what we have to always understand, what is the biological context to anything that we are discussing. If the same thing what Shritama is saying, if it is happening in liver, then maybe they want the same thing to happen. That's why cell is doing that. Otherwise, unnecessarily cell will not invest that. Activating multiple receptors is not going to happen unless cells need it, right? You will go and you will find always so many examples where either you have complementary pathways going on, or you have homeostatic balance over there. That's how the system will do it. Even in this case, the moment you have achieved certain level, there will be feedback loop. So the additional regulators will always be there that we are not going to talk about.
Things like different functionalities like E and B, which are different in function, is not expected to occur together.
No, they will occur, but then they will have different set of substrates. That substrate specificity will be determined by different.
Time not expected to occur.
Yeah, they will not happen. Yes. So that then that is driven by the ligand, no? Yes. I don't think it will be like that. The reason is, if the cell is having another signal, then it will respond to that. No, see, the ultimate effect depends again. That's what the way I understand is, if E and V are giving different biological effects, and both biological effects are necessary for cell to survive, both will happen. Okay. If A and B both are happening at the same time and both are opposing to each other, then cell will choose what is required at that moment. You understand what I am saying, right? Even if the signal is low, that's when the whole amplification is happening. Even if that's how the modulation will happen. No signal. I think something which we talked about. Whenever we talk about ligands which are present in extracellular environment, they are always in excess. They will always outnumber the receptors which are present on the membrane. Any time you measure it, you will always have excess ligands than the number of receptors. The number of receptors will determine what is happening. So you have limited number of receptors, then there will be limit of then that's when the amplification is happening. Many-fold amplification happens. The signal amplification. So again, so it's not the hand-wavy explanation, but the biological context is very important. At given time, what is required? That's, you can imagine, you take expression of CDKs and cyclins during cell cycle progression. Only at this specific phase, this CDK is there. Next one, it is moved. It's not only that they are degraded, no, you inhibit their expression as well, right? So this thing will happen even if both cells are coming, but what is required to do that, that is chosen. That's how the cell, what we can say, survive. The survival is very important. So based on the requirement and context, it will happen. Amount will never matter unless that biological activity has to be taken place. Is it clear? Okay. So let's finish this off. So in case when there is a decreased cyclic AMP, decreased glycogen breakdown will happen, which means that you have sufficient glucose in the system. Now you can synthesize. So there is this phosphatase. Now everything will reverse happen. So in this case, when you had excess, when you wanted to break down the glycogen, what you did is the phosphatase you inactivated. Phosphatase will do opposite function, right? Now when you have sufficient system, when you want to induce the glycogen synthesis, you activate the phosphatase, then it will inactivate everything. So that this phosphatase goes and dephosphorylase GP, which is glycogen phosphorylase, so that your GP is inactive. So GS will get activated, and then you activate GS, and this GS, so you have inactive GS which was phosphorylated directly by pKa. You put the phosphatase, phosphatase will dephosphorylate it, so that your GS is ready to this job. So again here the amount of glucose in the system is determining. If your glucose is low, this will be picked up. It doesn't mean that that's the same time there is no other pKa activation, but this pKa specific this enzyme will happen. And another context to this is that pK is not the only determining factor to get some signaling output. No. How should I say? I'll give one example. So let's say you have, I was giving example of Wnt pathway, right? Wnt beta catenin pathway. I'll just give one brief example. So what happens when pathway is inactive? Your GSK3 beta, so your beta catenin, which is the factor that goes and associate with the TCF transcription factors, is sequestered in the cells, right? But what happens? These kinase is phosphorylated. This phosphorylated molecule becomes a substrate for ubiquitination. In Hedgehog signaling, for example, which is required for our digit patterning and all, which happens to primary cilia. So I can give more better example of that. In glee transcription factors, whenever pathway is inactive, so whenever you have glee transcription factors, okay, whenever pathway is inactive, your glee transcription factor will get phosphorylated. And this is a good example because pKa phosphorylates this. Okay, this phosphorylation, so first GSK3 beta will come, then pKa comes, and then casein kinase comes. 3 kinases are phosphorylating. So you can understand the complexity now. pKa alone phosphorylation might not be sufficient to activate a substrate. There might be additional phosphorylations required. So this get phosphorylated. Then what happens? Whenever it gets phosphorylated, it becomes a substrate for a ubiquitin ligase called beta TRCP. These ubiquitin ligase can phosphorylate. Vaishnav, are you following this? Little bit. Good. If you have questions, we can discuss more. So when you have phosphorylated glee, it will become a substrate for beta TRCP, which will ubiquitinate it. And this ubiquitinated molecule will get degraded. This is pathway is inactive. When you act, so here also you have GSK3 beta. Here also you have pKa. In the same cells, pKa might be doing some other job only, but this pKa phosphorylation on this molecule will make only sense when this gets ubiquitinated by beta TRCP. Otherwise, nothing will happen in the cell. When you have pathway activated, phosphorylation doesn't happen. If phosphorylation doesn't happen, beta TRCP will not happen, which means that this molecule is active. So it's a combination of events that happens. So pKa alone is not going to do everything. His job is to go and phosphorylate. That's what it will do. But if that is the same time, if your GSK3 beta is not functional, then your signal will not happen. So additional regulators are there, that also has to be taken care. So what we are trying, it's not an isolated system, that's what I am trying to say. So, and it's not one-on-one system as well. So it's little bit much more complex than what we are right now thinking. Still you are not convinced? Okay. Okay, you have any more questions? So this is how the glycogen pathway is governed, and then we will talk about the amplification, and there is one bit which called as ACAP signaling, that is pKa molecules can act for localized signaling only at the nuclear membrane, or some of those examples will